This is the first set of notes on Unit 7. This particular set is about DNA structure. Uh, you know that DNA, of course, is the molecule that makes up the chromosomes that make up the instruction book for the cell. So that's kind of what the focus of this cartoon is, that DNA tells the cell everything else that needs to be done. The first section here is about the history of the discovery of DNA. So if you have not already read Chapter 10, pages 182 through 187, you need to go ahead and read that. And the notes that follow are just the high points about what happened. I'd like you to read about the various experiments that the different scientists did so that you have some understanding of what kinds of things have gone into the history of the discovery of the structure of DNA. So back in 1928, a scientist named Frederick Griffith discovered that if he was studying bacteria, and he discovered that if he had two different strains of the same species of bacteria. One strain caused disease, the other strain did not. And he took some of the killed disease-causing or pathogenic bacteria and put them in with the non-disease causing bacteria. And then he discovered that the ones that had previously been non-disease causing became pathogenic. So he figured out that there was some kind of factor that they picked up from the dead bacteria. He didn't know what it was. We know now it was DNA, but he knew there was some factor that caused the bacteria to be transformed into disease-causing bacteria from non-disease causing. Scientists continued to work on this, and a few years later, 16 years later, Oswald Avery discovered what DNA was indeed the molecule that caused this transformation to happen. He did this by doing various experiments to eliminate the other molecules like proteins that had been suspected. And he finally, 16 years later, confirmed that DNA was the molecule that caused the transformation. Scientists continued to work with DNA and viruses and bacteria. And in 1952, um, Albert Hershey and Margaret Chase discovered that they were working with a virus called a bacteriophage, which is a virus that infects um, bacteria. And they knew that the virus was composed of only DNA and a protein outer coat, and they knew that something in the virus caused the bacteria to become a virus making factory and make viruses until the point in which the cell died and released the viruses. And so they were trying to figure out what the factor was that caused the bacteria to become a virus making factory. Was it the virus's DNA or the virus's protein? So they tagged the protein and the DNA with different radioactive elements, phosphorus for the DNA and sulfur for the proteins, and then were able to test to see which part it was that became part of the new viruses that were produced, and they discovered that it was DNA that did this also. A few years later, well, around the same time, a scientist named Chargaff was studying DNA also, and he was checking to see which of the what the percentage was of the four different kinds of nitrogen bases that are found in nucleotides of DNA: adenine, thymine, cytosine, and guanine. And he discovered that regardless of what kind of DNA he studied, although different kinds of DNA had different percentages of these, the amount of adenine was always equal to the amount of thymine, and the amount of guanine was always equal to the amount of cytosine, regardless of what kind of DNA he did. We used that knowledge that he gained. That was one of the big hints about the structure of DNA. And so we talk about the base pairing rule, which we'll talk about later, and we name it in honor of Chargaff because he's the one that discovered this. And around the same time, 1952, in Cambridge, England, Rosalind Franklin was studying various kinds of molecules using a method called X-ray crystallography, which showed some things about the structure of these molecules. And so when she studied DNA, the picture that she took of the structure of a molecule showed a big circle with an X in the middle of it, which indicated that DNA was a double helix. So James Watson and Francis Crick, around the same time, were working on, there was a sort of a race, I guess, to figure out the structure of DNA. And so they used information from Rosalind Franklin and others and made a model and finally figured out using Chargaff's information and Franklin's information, they figured out the final structure of the DNA molecule as being a double helix. and showing the base pairs. Adenine always pairs with thymine, and cytosine always pairs with guanine. And so they are the ones that are credited with discovering or figuring out the structure of the DNA molecule. Now, it's important for you to know about all these scientists. You need to know their names, and you need to know what they discovered, but you don't need to worry too much about the years. I put the years on here to show you that this all happened in a relatively short period of time, and it wasn't really all that long ago. Yes, it was before you were born, but it was all around the time I was born. I was born in 1952, and so this all was discovered. The discoveries were culminated around the time I was born. So even though I may be old, I'm not that old, so really wasn't that long ago. So DNA, we know, is a nucleic acid. The name is deoxyribonucleic acid, okay? And it is a molecule that carries genetic information. When we look at cells that are not dividing, we see the DNA in the form of chromatin. Remember, the DNA is wound around a protein called histone and then twisted back and forth around itself to make the chromatin strands. You only see chromosomes when the cell is actively dividing. We talked about that in our last unit. And another way to describe DNA is a string of genes. A gene specifically is a sequence of DNA that codes for one protein chain. And we're going to learn in the next little bit how that works. So deoxyribonucleic acid is made of nucleotides. That's the unit of structure. Those are the monomers. Each nucleotide is made of 3 parts. It's made of a 5-carbon sugar, deoxyribose, a phosphate, and 1 of 4 nitrogen bases. The bases are adenine, guanine, cytosine, and thymine. This is adenine, and you can see here that it has 2 fused rings in its structure, okay, a pentagon and a hexagon that are fused together. That is characteristic of a group of nucleotides called purines, and that includes adenine and guanine. The other 2 nucleotides in DNA, cytosine and thymine, are what we call pyrimidines, and they have only 1 ring structure rather than 2. They have the hexagon structure but not the pentagon structure in their structural formula. So the 2 types of nitrogen bases, purines, adenine and guanine, have 2 ring structures, and the pyrimidines have only 1 ring. You need to know which ones are purines and that purines have 2 rings, and which ones are pyrimidines and that pyrimidines have only 1 ring. One way to remember this is using the sentence ags are pure, AG, ags are pure, purines. And that's just a simple way to remember which ones are purines. And if you can remember which ones are purines, the others are pyrimidines. Purines have 2 rings, and the pyrimidines have only 1. Now, the DNA molecule is a double helix. It's made of 2 chains of nucleotides side by side. Here's a diagram that shows a DNA strand. It just shows a part of a DNA strand straightened out, okay? Here's 1 nucleotide, the sugar, the phosphate, and the nitrogen base. And notice that there are lines that it's shown in between the paired bases. Adenine always pairs with thymine. Cytosine always pairs with guanine, okay? And notice the lines here, those are not really covalent bonds. They should be dashed lines because they're really hydrogen bonds that hold the complementary bases together. And so adenine and thymine have 2 hydrogen bonds that hold them together. Cytosine and guanine have 3. If you can remember C and G have 3, that will help you remember which one is which. You do need to be able to recognize these by the number of bonds and the shape of the nucleotides, the purines and pyrimidines. The two strands of DNA run opposite to each other. You notice that the pyramid shape here or the pentagon shape rather of the sugar looks right side up on one side and upside down on the other. That's because the 2 strands run opposite directions. They're called 3' end and the 5' end. Okay, notice that on this, and the numbers come from the carbons in the molecule. If you start with the oxygen and you'll go around clockwise and number the carbons 1, 2, 3, 4, and 5. The 5th one is sticking up over here on the side. Then in this side of the chain, the 3' end is down and the 5' end is up. And the other side is the opposite way. The DNA is read from one direction to the other. And so this side of the DNA molecule would be read this direction, and the other side would be read the other direction. This concludes the notes on DNA structure. The next section will be about replication.